Welcome. Today we're doing Lab 4, and it is about recombination, crossing over in a particular kind of fungus called Sordaria fimicola. Now, fimicola is interesting. One, it grows on poo. It's a dung fungus. And it has two genetic features that make it really ideal for studying the process both of meiosis and of crossing over in prophase 1 of meiosis. And the two things is that as meiosis goes along in this dung fungus, the spores that are created are held together in a structure called an ascus. And in this case, there's going to be 8 spores, and they're all going to be lined up in a little capsule, as I've called it, the ascus. Now, the thing that makes this really useful is that unlike any other organism I can think of, its meiotic products, its gametes, its spores, have variation in them. And there are 4 different colors that are possible in these spores. There's black, clear, yellow, and a kind of tan, sometimes called buff. So what that's going to allow us to do is to detect in a large sample the number of times that crossing over occurs, recombination occurs between one of these spore-colored genes and its centromere. In essence, you are going to be creating a map of the distance between spore-colored genes and their centromere. Now, here's how this is going to work. You have to take the fungus which grows as a haploid. These were grown on specific kinds of media, and then you can see we took a little chunk of the media and placed it buff, and in this case gray and gray. And so what's happening is that from this block, the mycelia are growing out in all directions. And when they meet, you can see there's a color boundary. And at that color boundary, the haploid hyphae find their mate, form a zygote by fusing their haploid nuclei, and then immediately undergo meiosis. And they create these 8 spore asci. And the 8 spore asci are contained within the little bitty black structures called perithecia that you can find piled up along the edges where these 2 different colors meet. In the lab, and I'll be showing you later, what you would do is you'd scrape off some of these perithecia. You'd place them on a clean slide. You'd add a drop of distilled water. You'd add a cover slip. And then you gently press, and you can sometimes even feel these black hard perithecia breaking open. And when you do it right, and the timing is perfect, you get an array of asci from the broken perithecia, and you can so clearly see the different spore color patterns in this row of 8 spores. Now there's 3 kinds we care about. There is 4 to 4, which indicates that no crossing over has occurred, and then there's 2 to 4 to 2, which indicates a crossover, and also 2 to 2 to 2 to 2, which indicates a crossover. So by counting up the number of asci that show recombination, 2 to 2 to 2 to 2, or 2 to 4 to 2, compared with the total number, allow you to answer the question, how often does crossing over happen between a gene and its centromere? And that's the data that you'll use to derive and create the maps that I'll assign to you.